Good morning. John Rubenstein just walked in so we can start the lecture. Morning, John. Sorry. I don't know why I had an impulse to do that. Sorry. Is this Mike? I'm kidding. Anyway, it's great to see everyone this morning. Welcome to the Distinguished Visiting Lecture Series, Department of Psychiatry. Thrilled today to be able to introduce our speaker coming all the way from Boston, Beth Stevens. Beth was an undergraduate at Northeastern, then went on to do her PhD at University of Maryland, College Park, and then saw the light and came west. Well, actually only partially saw the light, came west and landed just about 40 miles south of the appropriate target at Stanford. But did a phenomenal postdoctoral fellowship with Ben Barres, who's had an enormous impact on the field, focus on glia. During her postdoctoral fellowship, she made a really, I think, profoundly important observation for us as psychiatrists, folks interested in both psychiatric and neurological disease by finding that the classical complement cascade played an important role in synaptic formation, particularly in synaptic pruning. She's gone on now. We were just commenting that she is an assistant professor at Harvard Medical School, which just shows how broken their appointment and promotions process is. But I'm sure that is a highly temporary state. She's had a series of very important papers following up on this idea that the complement cascade system is critical in neural development. And then I'm sure most of you, if not all of you, know of some recent work applying this to the understanding of the genetics of schizophrenia, which I think is really having a tremendous impact on the field in a whole variety of ways. We're thrilled to have her here today. She is going to talk about synaptic pruning, I presume also to some degree about complement cascade's role in that, and then particularly its role for us in neuropsychiatric disorders. Beth.
Okay, thanks so much, Matt. This is obviously a huge honor to be here today and to be able to tell you a bit about our work, and also to come back home to the West Coast, which I have to say, despite the fact that I'm a native Bostonian, I sure do love it here. It's fantastic. Okay, so increasing evidence suggests that normal brain development is impaired in schizophrenia and a host of other neurodevelopmental and neuropsychiatric disorders long before the onset of the first clinical symptoms. And emerging evidence implicates immune-related pathways as well, but still many questions remain, including how could altered immune signaling contribute in any way to these disorders? What are the mechanisms? So we now appreciate that many immune-related molecules and immune-related cells, which I'll tell you about today, called microglia, these molecules are expressed in the healthy brain, where they play a role in brain wiring and brain development. We recently identified a role for the classical complement cascade in this developmental process called pruning, and recent work, more recent work, has implicated this immune cell that I'm going to tell you a lot about today, called microglia. So what I'd like to do is tell you about how we got into all this, in terms of what we've learned about how these immune pathways help sculpt synaptic circuits, largely based on work in the mouse visual system as our model. And then I'm going to move into how this normal developmental pruning process through these mechanisms could go awry in disease and disorders like schizophrenia, as well as neurodegenerative diseases like Alzheimer's disease. And I'm going to end a little bit with some implications and our efforts to try to better understand how to translate some of these basic findings into potential new therapeutics and biomarkers. Okay, so as you all know, the postnatal brain is remarkably plastic, right? We start off with far more synapses and synaptic connections than are maintained in the adult brain, but through a normal developmental process called pruning, a lot of these extra synaptic connections get pruned away permanently, while others get strengthened and maintained. Now, we know this process happens in multiple regions of the brain. There are different critical periods of pruning depending upon where you are. Sensory systems typically prune early. Now, in a mouse, this happens over the first few weeks of life. In a human, this can go on into many years, into the first few years of life. But in some parts of the brain, like the prefrontal cortex and frontal association cortex, this pruning process has been estimated to continue into early adolescence. And this is just a figure from Huttenlocher's work that just shows this example of this excess connectivity early on, and then if you look later, you can see that there's clearly some sort of pruning going on. So experience refines synaptic circuitry, and we also know that this process of pruning is necessary for precise brain wiring. So you can imagine if this pruning process doesn't occur properly, this could have long-term consequences both for circuits and behavior. And indeed, it's thought that pruning defects are thought to underlie schizophrenia and autism, neurodevelopmental disorders. There's some evidence for that. This is a classic study that I'll come back to by David Lewis's lab, where he looked in the prefrontal cortex of individuals based on autopsy samples and showed a sparsity of dendritic spines. Interestingly, in the deep layer, 2-3 pyramidal neurons in the frontal cortex, and that there was a decrease in these synaptic spines in some individuals with schizophrenia versus control. There's also some evidence that synapse loss and spine loss happens in autism. And so the other thing I want to say is that even though I'm going to focus today on the development, a lot of my talk will be focused on development, another interesting point is that synapse loss and dysfunction is a hallmark of many different neurodegenerative diseases, especially of the aged brain. And in fact, this has been best studied in Alzheimer's disease, where you can see also a decrease in spines and synapses that are thought to happen years before the first signs of pathology and impairment. And in fact, a lot of neurodegenerative disease involves synapse loss, raising the intriguing question that although each of these disorders are quite different with respect to its genetics, it's the part of the brain that it affects, could there be some common pathways, some common mechanisms that involve synapse loss? And if true, this offers a really exciting possibility that if we can find out what that pathway is, we may be able to perturb it and protect synapses in not just one disease, but many. Now, a major question in the field, and one that my lab has been intensely investigating, is how it is that during development specific synapses get pruned, because we know this process of pruning is not random. There's quite a lot of specificity to the process of which synapse gets pruned and which synapse does not. And this is a cartoon just to illustrate this point. So early in development, if you were to look, this happens to be in the visual thalamus, the system that we study, but this also could be happening in other parts of the brain. And the idea that initially this neuron is innervated by lots of presynaptic axons, lots of inputs, and they're about equal strength. And actually at this point in development early on, they're pretty weak. But over the course of development, you can see that there's a strengthening of some of the inputs, in this case the red one, and a loss of the blue ones, in this case the less active inputs. Meaning that the inputs that are less efficient at firing the postsynaptic cell tend to lose this competition, right? The idea is use it or lose it, and that somehow activity is setting up this local competition. Now that's been demonstrated in many systems. Neuromuscular junction, this is work from Jeff Lichtman's lab, Carla Schass and others in the visual system, cerebellum, this is climbing fiber. In all these cases is evidence that neuronal activity is critically important in this pruning process. But what are the mechanisms and the molecules that link changes in neuronal activity with the elimination of, let's say, the blue input but not the red one? And that's what we've been studying and focused on, is trying to understand the molecules and the mechanisms. Could there be, for example, molecular cues or tags that say eliminate the blue input but not the red one? Could there be protective signals that basically protect the red input from being eliminated? And once a synapse is destined to become eliminated, right, where does it go? Is it reabsorbed by the neuron itself, or could there be other mechanisms? Now, much of the field has focused, not surprisingly, on the neurons, because of course the neurons themselves are what's getting pruned. But emerging evidence in the field now implicate another cell type in this pruning and refinement process. And that cell type, of course, is glia, right? Glial cells, the non-neuronal cells of the brain that represent over half the cells in our brain. And only fairly recently have we started to really appreciate demonstrate that these cells do far more than just support the neurons. They're actively engaged in signaling with neurons, and one of the things we know they do is help sculpt and help the brain wire up and develop. There's been pioneering work from Ben Barris's lab, but many other labs now that show cells like astrocytes, for example, which ensheath synapses, release signals and actively regulate synapse development, structure, and plasticity. But I'm going to focus today on another cell, which back at the time this figure was drawn, it was this sort of brown amoeboid-looking cell off to the left. It's actually very sad to see this now. I'm almost embarrassed to still put it up here, but at the same time it shows progress because this wasn't that long ago. This is a review from Ben's lab, from Nikki Allen. This is just showing a beautiful real-life picture of an astrocyte in green and sheathing neurons. But again, I'm going to focus on this cell. And as you can see from this image, and you're going to show you a lot more pictures and images of microglia, they are far from brown and dull. They're incredibly interesting cells. They're incredibly diverse and dynamic, as I'm going to show you in a moment. But honestly, as a developmental neurobiologist, I've completely ignored this type of glial cell until about 5 or 6, or actually now about 8 years ago. And that is in part because we knew a lot about microglia, especially in the context of disease, because we knew as our resident immune cells, these cells are incredibly responsive to change, to injury, to disease, to inflammation. Reactive gliosis, reactive microgliosis in particular, is a hallmark of so many disorders. But much of the effort in the field is really focused on them as immune sort of surveillance, immune cells that respond to damage. They can be good guys and bad guys, right? So they can clear things like amyloid and dead cells, but they can also release neuroinflammation signals and things like that, which can then further contribute to various diseases. But one of the things that got our attention is, well, actually 2 things. One is, first of all, they represent about 7 to 10% of the cells of our brain. And until fairly recently, we thought that these cells came in after birth from immune cells, right? Like macrophages in our periphery kind of snuck in through an immature blood-brain barrier and took resident up into the brain and became what we now know as resident microglia. But very elegant fate mapping studies a number of years ago by Mary Murad's lab and Florent Genoux, they actually fate mapped microglia. They want to know where they came from. Well, it turns out they come from a special progenitor precursor in the yolk sac, and they're quite distinct and unique. These are tissue-derived macrophage precursor, but they're not the same ones that are circulating in your blood. They come from the yolk sac, and they make a pilgrimage into the brain in a mouse as early as embryonic day 8. So super, super early. In fact, they're the first glia in the brain. Before astrocytes and before oligodendrocytes, these microglia are in there. Now, we still know remarkably little about what they're doing in the developing brain, and in much of the field there's been sort of an explosion of interest in this. But I'm going to tell you a bit about their role after birth, although I have to say that we think that they could be playing a role much earlier in development as well. Now, the other thing that got us very excited about microglia when I started the lab was the fact that in vivo imaging experiments using a reporter line, this is a fractalkine receptor GFP mouse that was made by Stefan Young, which has been an incredible tool, and so much of discovery depends on new tools, and this was one of those game-changing tools. So these are mice in which all of the microglia are conveniently labeled with GFP, and so one can then put a mouse, shave off a little bit of their skull, and put a microscope over their heads, and just basically ask, what are these cells doing? What do they look like? And work by Axel Nimmeron and then later Wenbiao Gan's lab looked at these cells, and this is not their movie, this is something from Lasse Dissing in my lab, but what I want to show you is that these cells are incredibly dynamic, right? I'm hoping that I can play this movie for you. Is it not playing? Oh, that's not good. Sad. I hope the next one plays. No. Well, that's really sad. I'm actually going to just do something really quick, if you guys don't mind, because it would be unfortunate not to be able to play this for you. Take 2. Okay, so this is microglia, what they do, right? So all of their processes are constantly moving and surveilling the extracellular space, right? They're constantly doing this under normal, healthy conditions. Now, if you injure the brain, they also dramatically change, and actually they can recruit to the site of injury. Their processes can extend and retract. But I want to tell you that what got me excited about these cells is that they're doing this all the time. In theory, they're all doing this right now in your brains, especially when you're learning and paying attention to my talk. They're really dynamic, right? And so we started thinking about the fact that since they're surveilling, what are they surveilling? What are they interacting with in the brain? And we now appreciate that one of the things that they're surveilling are synapses, right? So this is a neuron that's also been transgenically labeled, so these are spines, these bumps, and they're constantly touching and contacting spines, axons, boutons all the time, right? And in particular, we wondered, could they be doing this during development? And because of the fact that we know that they're particularly phagocytic, ththese are our resident phagocytes, meaning unlike a lot of other cells in the brain, they're really good at engulfing things, right? We started wondering, well, could they actually be nibbling off some of these synapses? And maybe the way, if they are involved in pruning, that they're doing this is literally by plucking off and engulfing bits of synapses. And so we actually we developed a number of different assays and model systems to test this hypothesis. And one of the things we noticed, as I mentioned, and others had also observed this, that if you look in the brain during postnatal development, microglia were particularly phagocytic in regions of the brain that were undergoing a lot of pruning. So in the hippocampus during its critical period, in the visual thalamus that I'm about to tell you about during its critical period, in the cerebellum, and so on. And so the idea is, could they be actively remodeling these synapses? And this is an example of a microglia in the dorsolateral geniculate nucleus, which is the visual thalamus. You can see a green microglia interacting with blue and red puncta. These are the endings of the presynaptic terminals of retinal ganglion cells. And what we found is that almost every single microglia that we surveyed, if we then do 3D reconstructions, are full of synaptic inputs, right? So we can put cholera toxin in the eyes of a mouse that labels the axons very nicely, basically with this tracer. And then what we surveyed is that almost every microglia in the visual thalamus of a mouse is full of these synaptic inputs. And importantly, this is tightly regulated and developmentally restricted. If we look a little bit later, after this process of pruning in this window, there's a huge reduction in the ability of microglia to do this. They still do it in different critical periods, but at least during the period of very robust eye-specific segregation in this system, we found that they do it quite a bit more. So the question then becomes, how do they know which synapse to prune? Are they picky about this? Or are they just sort of eating up the debris after something else causes the pruning? And so we started thinking about the question that I raised at the beginning, which is that developmental pruning is very much regulated by neuronal activity, remember? And so we wondered if this process is sort of activity dependent and the less active input normally would get pruned, especially in the visual system where it's been really well studied, we wondered, could microglia be preferentially engulfing the less active input? And in the visual system, we can actually test this because we can manipulate activity. Now, the other thing we want to know, what are the molecules? Well, this brought back work that I had done as a postdoc in Ben's lab, where we unexpectedly identified a role for the classical complement cascade in developmental pruning in the mouse visual system. Now, this is a group of molecules, many of you are probably remembering your immunology, that this is a classic system that in the immune system helps to clear unwanted cells and debris, right? It's our first line of defense against an infection in the periphery. And what we found, quite surprisingly, through an unbiased screen in the lab, in Ben's lab, that this group of these classical complement cascade genes and proteins were very much expressed in this system by the neurons, the retinal ganglion cells, during the peak of pruning. And when we knocked out C1q or C3, these mice failed to prune properly in the visual system. So they failed to segregate into eye-specific territories, and they remain multiply innervated. And now in the immune system, as I said, its main job, these are a group of secreted immune proteins. We mostly think about this in the context of infection or disease, but what we found is that the brain, both neurons and glial cells, express many of these complement components, and these particular molecules like C3 are highly enriched during these pruning windows in the brain. Now, these are a group of secreted molecules, and the way it would normally work to eliminate, let's say, a bacterial cell is that C1q, the initiating protein of the cascade, binds to, let's say, that cell, and that initiates this proteolytic cascade that leads to the cleavage of the downstream components, C4, C3, CR3, for example, that ultimately lead to like the tagging of that cell or coding of that cell by C3. And one of the canonical ways by which that cell gets removed is through phagocytosis by circulating macrophages, right, in the blood. And so that's how it normally works in the immune system. But what we found is in the brain, these complement molecules were binding to subsets of synapses, and in fact, there was a sort of a subset of synapses being marked by these proteins, especially during development. And thinking about how this could be working, instead of the macrophages, we started thinking about microglia, in part not just because of what I told you about how dynamic they are, but because they're the only cell in the brain that express receptors for these complement components, including CR3, complement receptor 3. And so we wondered, could they be regulating this process? And so this is the model, is that these complement proteins like C1q and C3 are binding to subsets of sort of immature developing synapses, and that microglia, by virtue of these receptors that they express, if they see complement targeting synapses, that will initiate this phagocytosis process, very much akin to what happens in the immune system. And indeed, in support of that hypothesis, mice that lack CR3 in the microglia fail to engulf synapses the way normal microglia do. They're about 50% good at engulfing synapses. This is just measuring engulfment in that visual system assay I told you about. And if you also knock out the ligands, or the proteins like complement, C3 and C1q, there's also about a 50% reduction in the ability of microglia to engulf in this system. And moreover, and this is relevant to some of the consequences in the context of disease, what we found is that if you knock out the ability of microglia to do this, even by 50%, there are sustained defects in synaptic connectivity that maintain into the adult animal. And although we focus on the visual system as our model, because it's a very elegant model to study pruning, we have evidence now that suggests this is actually happening in multiple different brain regions. And so there's more synapses in these mice. And actually David Prince from Stanford went on in the C1q knockout mice, for example, and showed in the cortex they're hyper-connected, which is what you might imagine if you're not pruning enough, you have too many connections, and that unbeknownst to us at the time, the C1q knockout mice were more prone to seizures. So if you do EEG recordings, at least up through juvenile time, they were more prone to these absent-like seizures, and by physiology, there was more of a hyperconnectivity throughout different parts of the cortex. And this is really exciting to us because it suggests that this mechanism, if it doesn't happen properly, has long-term consequences in terms of functional and structural connectivity. And now we're running a lot of our mice through a lot of behavioral analyses as well, and this is all work in progress. Okay, but it also tells us that this can't be the only story, and we're actually exploring other mechanisms, and I'll give you a hint about how we're thinking about that in a moment. So back to the activity idea. So is it that microglia are just sort of secondarily engulfing these synapses, or is, again, how do we bring this together with that neuronal activity idea? And so we use the visual system once again as our model to test this, because you can manipulate neuronal activity in the eyes of a mouse. So you can block activity in one eye with injection of like tetrodotoxin or TTX, and you can set up this competition artificially. And then Dory in my lab, Dory Schaefer, asked, is there a preferential tagging as well as a preferential engulfment of the less active eye input? For example, the blue ones. And so she did that experiment. This is just a sort of a schematic of what she did. She blocked activity in one eye and then asked if the microglia are preferentially engulfing the less active or silent eye inputs. And then she did the flip experiment where she added things like forskolin, which increases the firing of that eye, and then asked if there was a preferential engulfment, in this case, of the vehicle eye. So in other words, set up the competition and ask if microglia care. And what we showed is in both cases, microglia preferentially engulf the less active eye input. And that raises many questions about how this might be working. In unpublished data now, we went back and repeated that experiment in the C1q knockout mice and showed that no longer, when you get rid of that eat me signal, that C1q, no longer do microglia care about less active and more active eye. That tells us that complement is somehow downstream or regulated by activity. And we're now thinking about mechanistically how this could be working. And I'm just sort of going to summarize this. I'm not going to show you all the data, but I want to just give you a sense in the lab of how we're tackling this question and why we think it is important. So one idea, since these are groups of secreted molecules that are expressed at the right time in the right place, one idea is that any synapse that gets tagged just essentially gets engulfed by microglia, right? Now that, we do have some evidence that complement is binding to the less active eye inputs. This is unpublished data. But that also suggests, importantly, that there must be some receptor or some protein that's specifically there at those less active synapses. Now that becomes really interesting and relevant later when we think about how to manipulate this pathway, right? So we're trying to figure out what those receptors or binding proteins are at synapses. However, we don't think that this is the whole story, because at first it's never that simple, right? And thinking about complement, and one of the things that kind of was hard to get our head around is the fact that these are secreted molecules. How is it that a group of secreted molecules could have such specificity? And the other observation is that when you look at microglia engulfing synapses, and we're actually now doing in vivo live imaging to get at this question, one of the things we notice is that they don't just continue to eat the whole cell or the whole axon or the whole dendrite. They seem to have a very preferential engulfment of bits of the cell, in this case the presynaptic terminal. And we have EM evidence to suggest that they're engulfing just the nub, the presynaptic terminal, during development. And others in the field have looked at this in the hippocampus, for example, and have shown spine engulfment. So how could that be working? Well, we started thinking about the possibility that there could also be a group of protective molecules that might work in concert with molecules like complement. And in this model, you can imagine that complement are secreted, they are binding to subsets of synapses, but maybe there's another group of molecules that essentially is expressed everywhere, especially on those more stronger synapses, the ones you want to keep. And that the synapse that lacks the protection, but also has the punishment signal like complement, that's the one that gets preferentially removed. And data in the lab now that I'm not going to share with you, but just sort of highlight some new work that suggests that there's a group of molecules classically known in the immune system as don't eat me signals. And one of the molecules is CD47. And this is a molecule that many of you may be familiar with in the context of pain and cancer. Irv Weissman, for example, has shown this is an immunotherapy, right? And what we found is in the brain, these are molecules that are expressed in the brain normally. And healthy cells and a lot of neurons have tons of these protective molecules on them. And what we think might be happening is during this process of pruning, there's a selective downregulation or redistribution of the protection that then allows the microglia and the complement to bind. And so we're now actively testing that hypothesis. But what we do know is mice that lack CD47, the microglia eat too many synapses, and they have the opposite phenotype as the complement knockouts. They have too few synapses. And so the idea of this yin and yang is something that's really appealing to us, because in the immune system there's a whole bunch of these protective molecules as well as molecules like complement. And that's how, for example, a macrophage knows not to go after a healthy cell or a self cell, because these are the molecular cues that tell that macrophage to eat me or not to eat me. And we actually think these molecules are working very similarly in the brain, normal healthy brain during development. And it also suggests the possibility, and this is Emily Lehrman who's been working on this CD47 story, that much like the immune system, these immune-related molecules are tightly regulated during development. And you would want to have that tight regulation, right? You wouldn't want to have these immune molecules overactive, pruning too many synapses or the wrong synapses. You'd want to have as much as anything the brakes in place as well. And so we're now looking at what these regulators are, what's turning the cascade on, what's turning the cascade off, and how we're getting at the specificity. So to summarize where we are right now, we've identified this as one of many mechanisms that regulate pruning. And by no means do we think this is the only pathway. Work by Carla Schatz's lab, for example, has identified another group of immune molecules, the MHC class I, also key molecules involved in pruning. Quite intriguingly, this is part of the adaptive immune system, yet these molecules are in the brain as well, regulated in a very similar way by activity. And interestingly, Carla's molecules and our molecules, at least the mice that lack them, have strikingly similar phenotypes, suggesting the possibility that somehow there could be some interaction with these immune molecules, and we're interested in understanding what those mechanisms might be. But the point is that these molecules and this pathway that I've described to you is regulated at the right time in the right place during these critical periods of pruning, and then it kind of gets downregulated via a variety of different mechanisms we still don't quite understand. But in the mature brain, there's sort of a downregulation of this pruning pathway. But what would happen if that pathway wasn't turned down, especially during development? And we think this might be what could be contributing to some of the synaptic and brain wiring defects in schizophrenia and autism. Now, this has been a hypothesis that you're familiar with. It's been out there for a while, this pruning hypothesis, but it's been very difficult to understand whether or not, A, does this really happen and does it contribute to schizophrenia, for example, and is the synapse and spine loss that, for example, David Lewis observed, is that really because of a pruning problem, or is it something else, secondary, right? And it's been really hard to get at cause versus consequence. Now, this is just a zoomed-in image of that image that we all show because it's really striking. This reduced spine numbers in schizophrenia, but it's not the only study. Of course, there's cortical thinning, evidence of cortical thinning that occurs in schizophrenia that is thought to be even at the early stages before the onset. And actually, Feinberg 40 years ago proposed the idea that altered cortical pruning could be important for the disease. But again, is it cause or consequence? Is it a loss or never formed? You know, we don't yet get this sort of chicken and the egg question. It's very hard to do that. Well, this is where, you know, genetics, human genetics can be incredibly powerful, right? And in the next few slides, I want to share with you recently published work by my colleague, Steve McCarroll, and his graduate student, Ashwin Sekar, who are using genetics to understand biological mechanisms. And as many of you know, a potential clue in terms of the genetic etiology was hypothesized to involve schizophrenia's strong association with genetic markers across parts of the human genome, chromosome 6, called the MHC, the major histocompatibility complex, or MHC. So the MHC contains what is, for common variants, a whopping effect, right? For schizophrenia risk, almost twice as strong as the next strongest signal on any other chromosome. It's the tallest peak in the Manhattan plot here. Now, this region spans actually hundreds of genes. Many encode proteins traditionally associated with the immune system. And this inspired all kinds of theories about immune mechanisms or immune causes. But no one's ever identified, you know, what are the genes that are really underlying this genetic risk and what the mechanisms are. And in part because this doesn't follow any mathematical or statistical patterns explained by any known genetic variants. So what could be mediating this effect? Now, I should say that, you know, Ashwin, a graduate student in Steve's lab, is one of these incredibly brave and brilliant graduate students who decided that he's not going to be shying away from a very hard problem. He zoned in on that region and for many reasons honed in particularly on a gene C4, which is going to sound familiar to you because it's a complement gene. It codes the complement component gene 4, which is right in this classical complement cascade that we've been talking about. Now, they were studying, Steve and Ashwin were studying extreme forms of genome variation in which a gene is present in many different structural forms in different people's genomes. And Ashwin had developed novel molecular approaches to quantify or measure the structural variation in different individuals, right? And it turns out that C4, if you look in this region here, C4 is actually two genes, C4A and C4B. And what they discovered was that there's incredible variation in the forms of different people can inherit, right? So some individuals in their genomes have extra copies of C4A, some have extra copies of B, some have neither. And what Ashwin did is just measure this to see how much diversity really is there. And it was actually quite striking. And so this led Ashwin to think about the possibility that maybe the variation and the variability in the isoform of the type of C4 inherits could underlie the risk. Maybe maybe this could underlie some of the risk in that part of the genome. And that's just sort of shows you an example in cartoon form of what I just told you. So the question that Ashwin and Steve set out to ask was, does variation in the C4 gene underlie schizophrenia risk? Now, this is a challenge because although Ashwin had developed, these novel molecular methods to analyze C4 structure in many different patients, it was very hard to then ask how this relates to schizophrenia because, of course, these are patients who have their DNA samples all over the world. And they came up with a very clever way to essentially make a map and develop ways to map how the structural variation of C4 mapped onto existing SNP data from data that was already available from tens of thousands of patients, right? So they built a map to infer the four most common forms of C4, C4A and C4B, and they essentially then made a molecular barcode based on the SNP pattern surrounding the C4 gene, and I'm just summarizing now, this work is published, but just to give you a sense of how they did this, then this allowed them to ask whether the C4 variant or form links to or relates to risk of schizophrenia. And this allowed them to take advantage of all of these giant data sets from all around the world. And what they found, in just summarizing the sort of take-home of the study, which sadly is very much oversimplifying a very beautiful study that's been recently published in Nature in January, but I just want to sort of give you the take-home message, which is that the alleles of C4 appear to shape risk in proportion to their effect on C4A expression. So here on the left you're seeing each allele's risk for schizophrenia. These are the four most common alleles. And you'll notice that a given allele shows the same level of risk regardless of whether the other variation in the MHC. And on the right side, what I'm showing you is how each allele generates schizophrenia risk in proportion to the expression of C4A. So the take home here is that the more C4A you make from the locus, the greater risk of schizophrenia. So this was a remarkable finding and discovery. But it still leaves us with this big question. How does C4A increase risk of schizophrenia? What are the mechanisms, right? What is the biology behind this? And this is where this beautiful collaboration sort of formed between Steve's lab and my lab, who's been studying complement in the context of pruning and development, and Mike Carroll's lab, who's an immunologist at Harvard, who's been studying C4 in the context of the immune system most of his career. And he's made some beautiful tools in mice. And all of our labs started getting together a few years back, have joint lab meetings every week, and we decided to come at this question from multiple different approaches, right, given our expertise and our backgrounds. And a lot of our students started interacting together. And so the idea was, well, maybe what's happening is that these complement molecules, that too much C4 could lead to too much pruning. And if so, the hypothesis was that this might be one of the mechanisms you might get over pruning. Can we link these two ideas together? Now, of course, we're not going to have an answer for you there, but I'm going to give you some data that suggests and supports this hypothesis. So the first thing we did is we got a hold of some brain tissue from brain banks at the Stanley Center, and we looked for where the C4 protein was, both in human neurons as well as in the brains. And we showed that, much like we showed in development, C4 protein is localized to subsets of synapses in the human brain. This is just an example of C4 in green and these little puncta that are co-localizing with pre- and post-synaptic markers. This happens to be in the hippocampus, but we saw this in a few other brain regions. We don't have quantitative data or power to ask whether there's more of this going on in schizophrenia versus control. This is all work that's in progress, but it tells us that C4, based on some nice antibodies that we validated, is there and is actually localizing to synaptic structures in the human brain. We also, I should say, in Steve's lab, they did some experiments. This is Heather's work, where she stained human neurons that were then differentiated and matured with these C4 antibodies and synaptic antibodies. And again, in these purified human neurons, also evidence of C4 binding to and targeting synapses, especially as those neurons mature. The other thing she showed is neurons make a lot of the C4, and that they actually secrete C4 in culture. You can actually measure the conditioned media and show C4 being expressed. So the big question we then asked is, does C4 contribute to or mediate synaptic pruning? And for this, we turn back to the visual system of the mouse, which is where we've been studying it, and using mice that Mike Carroll's made. And this is a work that was led by Alison Bialis, who was my graduate student and now a postdoc in Mike's lab. She did that eye-specific segregation experiment where you can look at refinement in the retinogeniculate system of a mouse, and she did it in the C4 knockout mice, and asked the question, does too little or no C4 lead to a failure to prune? That would be the hypothesis based on our complement knockout mice. So she assayed eye-specific segregation, the ability for the left and the right eye to overlap during development. And what she found is, first of all, C4 is highly expressed in the retinal ganglion cells during the peak of pruning, much like we saw, for example, for C1q, and that mice that lack C4 fail to segregate into these eye-specific territories, which you can actually do the these heat maps and look at the overlap between axons from the left and the right eye, and showed that these mice, there's actually a dosage effect, didn't segregate or prune properly. So this data together suggests or shows that lack of C4 leads to failure to prune, and now work in our labs is now going to test the hypothesis that too much C4, in particular because of C4a, could this lead to over-pruning. And so Mike Carroll, for example, has made these beautiful humanized mice that overexpress multiple copies of C4a. We're actually using viral strategies to do this in a very local way, in a regulated way, looking in particular parts of the brain. And the idea is, if we have too much of this pathway going on, especially during these critical periods, does that lead to over-pruning? And this is work in progress. We don't have the answer yet, but that's the strategy we're using these mouse models. So we want to better understand also what the difference is between A and B, why A specifically and not B increases risk. So we want to better understand that mechanism, and of course Mike Carroll's been studying that in the context of the immune system, and we're trying to develop strategies to test that mechanistically in terms of its preference for synapses, its binding proteins, things like that. And we also, in my lab, are now moving out of our comfort zone of the visual system and moving into the frontal cortex. And we're trying now to map out when and where pruning happens in a mouse visual system, in the mouse prefrontal cortex, and then using these approaches to ask about pruning and over-pruning and what the functional consequences are in terms of connectivity and also in behavior that may be relevant to schizophrenia. And we're also moving on to other models in parallel to this, including non-human primate models like marmoset, and also stem cell-based models like organoids, to test this, because now we have a genetic handle on this. We can start to manipulate this pathway. And others are also, a student from my lab is also looking at another gene that's been increased risk of schizophrenia that also seems to be potentially converging on this pathway. And so now using these strategies, we're trying to get at this hypothesis. And the other thing that I want to sort of end with here before I move on to the last part of my talk is bringing back this question of, could pruning help us to understand the age of onset of schizophrenia, which is in adolescence and early adulthood. We are very interested in knowing whether there's a developmental process that happens earlier that can later be revealed during adolescence, or is there something happening at that critical period because that's the last part of the brain to prune. So for example, might pruning become too intense or prolonged in some individuals, and maybe that's what's causing the timing and the risk. Might pruning expose other pre-existing vulnerabilities? We're very much interested in testing the idea of a double hit, where you have a genetic hit, like let's say C4a, and then you can then expose these models to another hit, let's say a stress or some sort of immune challenge, and then ask how that impacts the circuit and also the system. And we basically are very much wanting to dig in and better understand what is changing in the adolescent brain, because we still know remarkably little about this. And then we're hoping that this can allow us to develop better models to test these ideas. So in the last part of my talk, I'm going to sort of just segue into another thing I hinted at the beginning of my talk, which was that, one of the challenges with schizophrenia and autism for that model is that if we really want to get at some of these hypotheses, it involves us we need to be able to develop better models to test this idea. And there aren't right now any really robust animal models to study things like schizophrenia, which is what we're trying to, and others in the field are trying to develop. But synapse loss is also a hallmark of many other adult onset diseases, including Alzheimer's disease. And this is a hypothesis that we put out there even when I was still a postdoc in Ben's lab. The idea that a normal developmental pruning mechanism, which normally gets shut down, could become reactivated aberrantly in the adult brain, and that could increase risk and cause synapse loss in these sort of age-related neurodegenerative diseases. In other words, could this developmental virtue become a neurodegenerative vice? And if so, could this pathway I've been telling you about today be one of the mechanisms by which this works? Now, there's evidence that microglia are clearly at the heart of a lot of neurodegenerative diseases. We've already mentioned that. They've become activated. They're part of sort of the inflammatory side of the story. But what about early, before all that happens? And in particular, particular, I think there's a lot of evidence in Alzheimer's disease that synapse loss happens quite early in the context of the disease, both based on human work as well as animal models, and that synapse loss is the strongest correlate of cognitive decline in Alzheimer's disease. And so this led us to wonder, could this pathway be happening at this very early stage, before inflammation, before plaques, before tangles? And if so, if we perturb it, if we block it, can we protect synapses and ultimately some of the cognitive decline? And so we know from the work of many that complement, not surprisingly, is upregulated. It's localized to plaques. This, again, is thought to be sort of late-stage disease. And we also know from the work of many that activated microglia are prominent players in Alzheimer's disease. They surround Alzheimer's plaques. They're good guys and bad guys. They help clear plaques, but they also can promote inflammation and things like that. But we wanted to know, and just sort of cut to the main question here, does microglia and complement, could they be contributing to this very early and region-specific synapse loss prior to these sort of like pathological hallmarks like plaques? And why is this relevant to schizophrenia? We're going to come back to this, because we think from the work of our lab now and other labs, including Eric Huang's lab here at UCSF, that this pathway I've been telling you about today could be representing a common mechanism of synapse loss. And if we could really better understand it, we might be able to harness that information to think about therapeutics, right, that may then be someday relevant to schizophrenia and other disorders and neuropsychiatric disorders. Now, there's also genetic evidence that implicates both microglia and complement in Alzheimer's disease, TREM2 especially, CR1, complement receptor 1, ApoE, ApoJ. All of these are genes that have been linked to Alzheimer's, but people really haven't been thinking a lot about mechanistically what that means. Now, what we set out to ask is, could early synapse loss in Alzheimer's be mediated by an aberrant reactivation of this normal pruning mechanism? And this is work spearheaded by Soyeon Hong in my lab in collaboration with Dennis Selko and Cynthia Lemire's lab. And we took advantage of the fact that there are actually some very good, decent animal models of Alzheimer's disease, and that we could then use these models, which the field's been using for years, to rewind the clock to the very beginning and ask, could this pathway be happening early? Could it be regulating and mediating early synapse loss? And if so, how can we understand mechanistically how to prevent it? And does that prevent synapse loss? And what we found is that synapse loss in the Alzheimer's models happens very early. We know exactly now when and where this happens. It happens in the hippocampus. And it happens in these mouse models prior to any plaques. It happens at 3 months in many cases. But it doesn't happen everywhere, which is one of the points I want to make, because specificity is one of the most interesting parts of some of the work that's coming out here, is that it's not happening everywhere. It's happening in vulnerable brain regions. In this case, it's the hippocampus, and it's happening quite early. And what we found is when we looked at the brains of these Alzheimer's mice, we saw a striking upregulation of complement in these vulnerable brain regions. This is dentate gyrus and frontal cortex. But notice we didn't see this in striatum and cerebellum. And we saw this actually in several different mouse models, that there's a region-specific early upregulation of the complement, and that when you zoom in on that, this is just a quantification of that data, when you zoomed in, we saw that very much like development, these complement molecules were targeting or tagging subsets of these vulnerable synapses in these vulnerable brain regions. And so this then allowed us to say, now that we know when and where this is happening, can we start to manipulate the pathway and protect some of the synapse loss? And so what we asked is, if we then knock out C1q, C3 in these models, what are the consequences? So we crossed, in this case, in Alzheimer's mice, this APP-PS1 mouse to a C3 knockout mouse. Then we looked at synapse loss as well as cognitive dysfunction in the deficient, C3 deficient mice, as well as the Alzheimer's mice. And what we found, and this has now just been recently published, is a significant protection of the synapse loss, which we can quantify with these synaptic markers in the C3 deficient mice. This is a early stage animal, so a 3 to 4 month old mouse, but our collaborator Cindy Lemire has gone on and showed in a 16-month-old mouse we can still see a protection of the synaptic loss. And most interestingly, and this is unpublished work, she showed that mice that lack C3, these Alzheimer's mice, they have improved spatial and contextual memory performance in the older mice. And most interestingly to me is that in these mice, despite the fact that there's protection of synapses and also in cognitive function, they have more plaques. And this is an important take-home because people have honed in on the plaques as a readout, but I actually think given the fact that cognitive decline and synapses are more correlated, that they may be looking at the wrong thing and they may be looking at the wrong timing. So this tells us that there may be a critical window in which to manipulate this pathway. And so we want to know, I'm going to skip a bit about the mechanism, but I just wanted to just give you a highlight here, is that we know from the work in the Alzheimer's field that there's a mechanism that's been identified, which is this A-beta, not the plaque, but the secreted form of A-beta, these oligomeric A-beta species. And if you add that onto a hippocampal slice, you can see a profound decrease in synapse loss. And so we wondered, since Alzheimer's mouse, since A-beta has been shown to be synaptotoxic, could C1q and A-beta be working together to mediate this early synapse loss? And what we found is indeed we can recapitulate the synapse loss in a wild-type mouse. We can inject this synaptotoxic A-beta into the ventricles and then assay synapse loss on the other side of the brain. And then we can manipulate complement and then see if we can protect that synapse loss. And much like the genetic models, we see a striking upregulation of C1q when you do this in terms of the A-beta injected mice. They go to synapses, much like we saw in the genetic model. So this is now a model to test this sort of acute synapse loss idea. And importantly, when we look at synapse loss, we can see a robust synapse loss a few days after injection of A-beta. That then allows us to then say, if we block C1q, can we rescue the synapse loss? And what we found is that A-beta-induced synapse loss is rescued when you do these experiments without C1q. So if you look at C1q knockout mice, we no longer see the synapse loss. And that tells us that they are somehow working together. And this is very exciting because we found a way and a potential mechanism by which A-beta mediates the synapse loss. But it also then is starting, we have now a model system to start to test novel approaches to block this pathway, right? And so we use this new antibody. This is a novel C1q blocking antibody that was in collaboration with Ben Barres and Arnold Rosenthal, who are part of this Annexon Biosciences. And we treated this mouse with this blocking antibody that essentially blocks the function of C1q by preventing the cascade activation. So it's a way of, in a very acute way, of manipulating the pathway. So Soyeon injected this blocking antibody into that same model and then added A-beta. And what she showed is that anti-C1q antibody rescues the synaptotoxic effect of A-beta in this model. And we can also show that by doing some slice physiology. It also rescues the A-beta-induced LTP induction and some of the other assays that have been done in this context. So bringing it back to microglia, we wanted to know then if microglia, are they aberrantly pruning or eating these synapses in this model? Again, without any plaques, which is why we developed this non-plaque model. We wanted to know, are the microglia responding to this? And we took advantage of that same kind of assay where we can measure engulfment of synaptic elements. And what we did is measured synaptic engulfment in mice that had the A-beta versus control. And what Soyeon showed is that the microglia indeed are overeating synapses in this model, and that when we block CR3 or complement, she sees a protection in the engulfment of the synapses, as well as more synapses. So this brings the same exact pathway we've been studying in development to play at this early stage of this Alzheimer's mouse model. And so what this is telling us is that this pathway that we've been studying in development is happening early, it's happening in a rather specific way, and that we've now shown that multiple different ways to manipulate it can protect the synapses, and we think some of the cognitive decline in these models. There's many questions that are relevant not just to Alzheimer's but to schizophrenia and a lot of the other, because now we have an assay to start to look for mechanisms. For example, we can ask, what is complement binding to its synapses? If we could figure out what that molecule is, we could try to figure out a way to block the specific binding of synapses to complement and prevent potentially the synapse loss. We also want to know what's turning it on and what's turning it off. And we also want to think about ways to validate this in human tissue, right? So we're looking in Alzheimer's models as well as other disease models, and we're thinking about the possibility that this might also represent an early biomarker. We can get CSF samples from preclinical Alzheimer's disease patients, for example, and see if this is changing at the earliest stages at the time where synaptic loss could be happening. And in addition, and this is my last slide as a take-home point, is that although I've told you about Alzheimer's, and some of you are wondering why do I care about Alzheimer's disease in terms of schizophrenia, we actually think that more evidence is coming out now that suggests that this pruning pathway is aberrantly activated in a host of different diseases. Not just Alzheimer's disease, but work over the last couple of years has implicated the same pathway also in glaucoma, frontal temporal dementia. This is a beautiful paper by Eric Wang here at UCSF. And unpublished data from my lab suggests that this pathway is turning on again quite early in a very region-specific way in different models of Huntington's disease. Again, suggesting a circuit-specific vulnerability and that something is triggering this pathway to come on. We don't know what that trigger is yet, but we're obviously very intensely trying to understand what that is. And then the idea is that if you can then block it, and in all of these cases we've shown evidence of blocking the pathway either genetically or with antibodies, it seems to be able to prevent some of the synaptic loss. And what we're now doing is asking what the consequences are for disease progression, cognition, behavior, and those all work in progress. So I just want to end by thanking a lot of people from the lab, past and present, really incredible collaborators and funding, and most importantly my lab, who are just an amazingly talented and excited group of people that get me excited to come to work every day. So thanks so much. I'm happy to take questions. Yes?
So Erwin Feinberg, his hypothesis is also interested in slow wave sleep over the course of adolescence, that it was related to pruning. I'm wondering if you or anybody have looked at the slow wave sleep.
We haven't looked at slow wave sleep. However, we have a project in my lab now looking at whether or not there's a link between sleep and pruning, just normally, in particular using some of the pathways we've identified. And if we can see if that's true, if there's actually a remodeling or pruning during sleep, we can then start to think about how that might relate to some of these disorders. And so I think there's sort of this basic question of whether, why do we sleep? What could be going on there? And based on the fact that activity is also a key role here, our hypothesis is that this may be happening during normal sleep, right? So we're testing this in various models now. And then the hope is that this might then overlay into schizophrenia and some of these other disorders. It could be a very interesting potential hypothesis, but it's still quite early.
I mean, it'd just be interesting to see if your sleep-wave knockouts actually have more slow wave sleep.
Exactly.
And the hyper, the older
right. Exactly. So we're running these mice through a lot of EEGs now. We're trying to just sort of characterize sort of baseline EEG activity and potential sleep, changes in sleep in these different mice. And so we're just at the beginning stages of that. And so we'll see. Yeah, great question.
So you emphasized that this is a developmental pruning process. And in talking about the Alzheimer's work and other work, you identified this as an aberrant reactivation of this developmental pruning process.
Yes.
But I'm wondering, we talked a bit about this last night, but I'm wondering what we know about this particular set of mechanisms, molecules, in the process of synaptic remodeling, distinct from developmental pruning.
Right.
And particularly as we start to think about, well, can we arrest neurodegenerative disease processes by, blocking this? Is it possible that the expression of complement proteins also play an important role in plasticity?
Absolutely. There's some evidence that this might be the case. We've focused much on just developmental pruning just because the models and everything's there. But now we're moving into asking about learning and adult plasticity in hippocampus and other brain regions, because these molecules, they're still around. I mean, they're expressed. I would say in the case of microglia, they may not be as phagocytic in the adult as they are during development, but in no way does that mean they're not actively, dynamically interacting with synapses. So there's some evidence, at least from Ben's lab, Ben Barris's lab, that the complement C1q deficient mice have defects in LTD and plasticity in the hippocampus that seems distinct from the developmental. We have other evidence unpublished that suggests the same. So we're just starting to understand that, and part of what we need to do, and why we've sort of held off a bit on this, is to separate out development from adult plasticity. We have to make new tools. We have to be able to knock this out and manipulate complement in the right time and the right place. So now we're making conditional mice, inducible mice, and using viral and other strategies to turn complement on or off in the adult, independent of normal pruning. And I think that's going to be a really important question, and it's very relevant to schizophrenia and some of the questions I raised there.
So just a quick follow-up. In having plasticity, synapses strengthen with activity, and that's very much a process that seems to be playing a role in deciding which synapses are in developmental pruning. Is there any evidence yet of differential expression of any of the complement proteins you're looking at in synapses that have been subject to LTP, classical LTP induction?
Yeah, so we're not definitive evidence of that yet. What I can tell you is that we have a paper that are now just pulling together a series of observations we made a number of years ago, which is one of the regulators of complement expression, and now we think localization as neuronal activity. And that C1q in particular is really transcriptionally regulated by neuronal activity, and almost if you fire retinal ganglion cells, stimulate them various ways, we see an increase in the transcript within 15 or 20 minutes, like really rapid. And so we also have evidence in the visual system that with experience, like dark rear a mouse and then open their eyes and expose them to light, there's this huge upregulation of C1q in the visual areas after experience. Evidence is now suggesting that these, both the complement proteins and molecules themselves are regulated by activity, as well as microglia. And so now the question is, like, how is this really fitting into plasticity models? And so we're doing this now in terms of inducing LTP, either chemically or by electrophysiology, and seeing how that changes. And we're trying to generate new tools. Where my graduate student is now doing this, where she wants to get a tagged version of C1q or complement, where you could then watch where it goes in the context of spines or synapses that are stronger versus weaker. These are like the types of things we're working on. It's just not, they're just hard experiments, but we'd love to know the answer to that. Yeah. Sure.
Talking about the monozygotic concordance rate of whatever the 50% in schizophrenia and support for the two-hit hypothesis. And I'm curious if you can speculate on that little, how is this, we think maybe that hit happened natally or early in development.
Yeah.
The system obviously undergoes some kind of hysteresis where there's a memory of that insult, where that memory might be encoded that affects the complement system, pruning perhaps 10 years later.
Right. Right. I'll tell you what we're thinking about, and like a mechanism that to us is testable in our models, right? Which is, especially now that we have these mouse models where we can overexpress either C4a or we can manipulate this other molecule or this other gene, where we can now give the genetic risk, and then we can then take those mice and then expose them to either prenatal or MIA, this maternal immune challenge situation, just one hit very early in development, and see how that impacts pruning and the circuit development. And then we can also do a later hit, let's say during this window of adolescence, which is something we're also very interested in. And then ask how that challenge could, if it does impact synaptic pruning and synaptic connectivity, and if so, does it happen through our mechanism? And one attractive idea in my mind is that microglia are incredibly responsive to these immune-related challenges. It could be stress, it could be infection, it could be a peripheral challenge, and that they are really kind of, well equipped to not just respond to, but potentially be changed. And so one idea that my Bostock testing is that if you then expose a mouse to a challenge early, how does that impact microglia development, function, and its ability to interact with synapses and neurons, not just there, but throughout life. And so we're doing a lot of those experiments now, and I think other people are also thinking about this, and also complement. So again, there's lots to be done there, but I think we're now in a position where we can start to ask that and also figure out when's that critical period, and then what the mechanism is. And then, because we have tools to manipulate complement and microglia, we could then ask, if we knock out a pathway only in those cells, can we rescue some of the changes? Yes.
So, fantastic.
Thank you.
Thank you so much. Like, incredibly clear and great.
Thank you.
I want to sort of follow up on that idea about hits. And think about maybe the difference between sort of thinking about hits from a biological standpoint and thinking about hits from a kind of pathophysiological or genetic risk standpoint. So if you have a first-degree relative with schizophrenia, you go from a population rate of about 1% risk to 10-fold that, right? Maybe 20-fold. So you go from a 1% risk in the population to 10 or 20% risk. If you carry nothing else but the variant that you described for us, you go from a 1% risk to a 1.27% risk of schizophrenia. And that's only with the highest risk allele. If you look at the schizophrenia genetics literature, there are over 100 loci. You need to be in the top decile in order to begin to get close in the population to what the genetic load is if you have a brother or sister. So what that suggests is that from a genetic standpoint, based on the biology, it's not a two-hit problem. It's a 90 to 100
multiple hit problem. Yeah, definitely.
And so then I guess so what I'm wondering about is and I think this is not just reflective of this work, but in thinking now about this important step of moving from genetics to understanding pathophysiology. How do you think about sort of effect size in terms of understanding whether or not, sort of where you're looking at this kind of cause and effect, driving pathophysiology versus sort of moderating effects on the periphery of some sort of driving etiological factor?
Yeah.
So how are you thinking?
Oh, man, that's a really hard, you should be asking Steve McCarroll that more than me probably, but I can tell you, this is a hard problem. But I can say that if there was a way, this may not really directly get at your question, but if we could figure out a way to stratify our populations, let's just say there was a biomarker, some indication, let's just say complement was changing in the CSF, or there was other evidence where you could then take the C4A individuals, and then there was some way then to stratify the population to start to test some of these ideas. Whereas if it's not done that way, it would be very difficult, I would think, right? And I think there's clearly a need also to get at the biomarker aspect of this. I don't know exactly how one goes about this, but the other thing that we talked about at dinner, which I think is also really relevant here, is the idea that there could be converging biologies that involve multiple genes that could be converging on, let's say, pruning pathways. Now, if that were true, pruning may not explain all of it, but if there were 4 or 5 or 6 different genetic variants or hits that impact in some way a pruning-related pathway, that's something that one could then start to think about. And so we are thinking about that in the context of other genes, and we have another one that also may be related, and maybe more and more of these synaptic-related genes might be a way of thinking about that. And that way you're not looking at 1.2%, right, or folds.
So I guess we have time, so I get to follow up this prerogative being the chair.
Yes.
So I get so that makes total sense to me, and I think particularly this idea that there's convergence could be really helpful. But I guess the other question, and it may relate sort of where you're headed in Alzheimer's, is that so let's just say that what's happening is that the tenfold increase in risk is something else, and then you have this slight increase in risk from complement, but you begin to find that there may be other molecules that are related.
Right.
I guess I just wonder the difference between thinking about sort of therapeutic traction with that it may not matter in some respects what the overall driving etiological problem is. If whatever's leading to this initial problem with Alzheimer's, it may be that what you are beginning to find may be an insight into resilience, right, as opposed to a major insight in risk.
Right.
So I just wonder whether or not that's sort of also kind of the flip side of thinking about how do you collectively get enough risk to model. Maybe the question is not that. Maybe
but the protection.
Yeah, how much traction do you have, and can you turn this on its head and start thinking about manipulating the pathway as a resilience kind of idea,
Yeah.
as opposed to...
You know, I think that's I think people don't think about it in terms of protection, but it's an incredibly interesting question. And our hope, too, is that Alzheimer's may give us a bit of a better handle on some of this. And also there's models that you can use that we're ahead there. And so, I mean, and also putting all this together, I mean, if you think about it, like, there's no A-beta link to schizophrenia or Huntington's, completely different gene.
Right.
To me, one of the things that's most intriguing about this is that different things can turn this pathway on. Genetics, environment, multiple things. But in the end of the day, once it goes, it's going in a pretty circuit-specific, region-specific way, which gives us a window of opportunity if we can figure out when the vulnerable circuit starts to go, which in the case of Alzheimer's, if there was a PET ligand, or if there was a synaptic biomarker, or if there was something that's going to tell us about cognitive decline versus just everything grossly, then we can hone in and start to think about manipulating the pathway. And that alone might not be the only thing either, but if we could slow it at the right time, that might then someday, if you think about even schizophrenia, the prodrome and looking at risk, and then finding again a way to say, well, these patients we might want to image. There may be ways. Although I will say that the idea of manipulating and mucking with pruning an adolescent brain is still kind of scary, as you're probably thinking about. You know, in Alzheimer's, you know, we're past that critical period, and you want to protect too many of our synapses when we get old. But when you're an adolescent and you're still, in theory, undergoing remodeling, this idea of tweaking something, unless you know what you're doing, is rather scary. So I absolutely, no way would we imagine jumping into therapeutics for schizophrenia. But I think the idea is that if we can figure out how it's working, and that's one of the reasons why we're really honing in on, like, what's the receptor? How do we get specificity? If we could get a small molecule that specifically disrupts that synapse, that target, that pathway, then it won't be so scary in the sense that you could get at specificity. And in the end, that's what we all wanted to do. And we hope that something like Alzheimer's and Huntington's might give us a little bit of a handle on that someday might be relevant when we know more about schizophrenia. Yes?
She gets okay.
I don't know who I'm supposed to be. Go ahead.
So just to Matt's point, one idea that I've been thinking about for a while now, in a way, is driving some of the work in the human schizophrenia program research. There is emerging evidence from a variety of methods in humans that patients with schizophrenia don't strengthen in response to plasticity inductions in the way that helping with the plasticity mechanisms may not work. This comes from things like direct current stimulation and electromagnetic stimulation. And one of the things that I haven't heard you specifically talk about, but it sort of supports this idea of a different mechanism that may converge in a way on the same pathway. To the extent that LTP, for example, is dependent on NMDA, glutamate neurotransmission, and there have been a number of suggestions of dysfunctional NMDA receptor activity in schizophrenia. Is it possible that this could interact with excessive, complement expression? Yeah. That they're also not strengthening their synapses normally. Experience, which could converge.
Totally. Absolutely. I mean, I think in the end, again, things that weaken, activity-dependent mechanisms that weaken synapses, which again in development, if it's done right, is exactly what you want to happen. But if it's not quite right, what you almost could have is these sort of senescent synapses that are just not working properly. And this pathway might even be tricked to say, well, let's get rid of it. It's not supposed to be there. And that might even homeostatically be a good thing initially, but then I think the process can get out of control. And so again, figuring out, a lot of the lab is really focused in on this really basic mechanistic questions, thinking that we really better understand when you're competing, two inputs are competing, and one's weakening, which we can induce, and the other one's not. What are the molecular cascades going on in that input? We're doing that, actually. We want to know what's upstream. We think it may be a calcium-dependent, caspase-dependent process. What's then happening and revealing itself on that less active buton or spine? And then how is that setting this whole thing off? And it could be that many other things are being set off by that as well. So in the end, that's upstream. And it's really relevant to this activity being an upstream regulator. So we really want to better understand that. So it's a great question. I wouldn't be surprised if it converged in some way on the NMDA receptors. Yes?
Hi, this may be an example where a little knowledge is dangerous. My understanding is that there isn't evidence of reactive gliosis in schizophrenia. Is that right? So how would you explain
Well, honestly, there's just mixed evidence in the sense that there's just not a lot of careful analysis on glia in schizophrenia, and maybe other people in this room can speak more specifically to this. But one of the challenges we have with microglia, and this is something that the field and my lab and others are really working on, is that we don't really have a good marker of reactive microgliosis. We can see them really angry if something really bad happens, like if you injure or have something really dramatic happen to the brain. They get bushy and they get kind of angry looking. They express some markers, but the ones that are potentially not working properly, we don't really know that yet, unless we do these types of assays I described to you today. So we need a better molecular fingerprint, if you will, of what microglia, when they're not behaving, are doing. And a lot of people are doing that now, profiling, both single cell, proteomic analysis. And what we're seeing is there's actually a whole range of states a microglia can be in. And to my knowledge, we just don't have an antibody or a convenient marker that we could stain a brain and say that microglia is not behaving, unless it's really dramatic. So when you don't see something dramatic, it doesn't mean they're not misbehaving. And I'd say the same is true for astrocytes. And I don't know, there have been studies that show some evidence of microglia activation in various ways, and also transcriptional data where they profile and they see changes in these gene expression of the glia genes, but not careful analysis to show dramatic changes in microglia that I know of. And I don't also think there's been a lot on the PET imaging side either, although someone here might know more. And the other thing we're trying to do is develop more sensitive PET ligands that gets at, again, these changes in microglia that are happening that can tell us more about its biology versus just inflammation. And so maybe when we get those tools developed, and this is all happening in the field, we'll then be able to go back to the same samples and the same data that's out there and get a better sense of when and where microglia and astrocytes change. That's what I would love to be able to do. But right now I don't think we have the markers, unfortunately. Yes?
Assuming the complement system was co-opted early in evolutionary history for neuronal synapse remodeling, pruning, etc., it makes sense to me that the effect size of any one player would be small, right? Because it's genomic balance.
Right.
You wouldn't want an infection to completely upset, throw off the trajectory of the brain development.
Right.
So I guess that makes me think, what's the simplest organism in which we know microglia and the complement system are playing a role in synaptic pruning? And is there any memory of infection or inflammation that can be measured or assayed, followed in that kind of simple model organism?
Yeah, we've been thinking a little bit about that. I mean, Drosophila doesn't have a classical microglia or complement, but they have those immune-like molecules that could be representative. I mean, so that's why mouse has been a pretty good model for this, but it's not particularly high throughput, and it's challenging in other ways. But I think you're onto something. I mean, maybe it could be zebrafish and other models like that might be helpful. To me, I think we've come this far in mice that we actually are pretty well positioned to start to ask some of those questions, just because, at least in the mouse, we know a lot about when and where all this stuff's happening, and we know that this pathway is involved. And I think we could start to get at this memory question, as we just talked about, in different ways. But in the end of the day, though, we do want to know, is it relevant to humans, right? So a mouse is a mouse, and it's not going to tell us all about the mechanisms of schizophrenia. But if we can then hone in on a pathway and a mechanism, then the idea is to go to some of these other models I mentioned and start to test this in multiple different ways, and including some of the human data and human patient material that together might point towards this mechanism.
You may not see him in the back, but John.
Hi, John. That's a good question. Yeah. I don't think it's known. This is complement deficient people, if you're asking me. Yeah, I don't know if it's been shown in terms of how that relates to risk to any of these diseases, but it'll be really interesting to ask. The question someone almost always asks me, especially if I give a talk to an audience like you, is, well, do complement deficient people have cognitive problems? I don't know, because it hasn't really been looked at super carefully, and there's probably some redundancy in the complement cascade. Clearly lupus and some of these autoimmune, there's cognitive issues. But in terms of whether there's developmental issues, I don't think it's been carefully looked at with enough power that we would know. But I think it's another question that's be really worth looking at. now that we know more than we used to know. Yes?
Tony, get the last word.
Thank you very much. I do functional MRI research work in adolescent depression, and I found your talk extremely interesting.
Thank you.
The question I was wondering is that depression is something, obviously, that you're developmental, it's in adolescence, affects the hippocampus, it's important. I was wondering if anyone in your lab or any other lab looked at depression.
Yeah, we have not, and I'm not sure if anyone's really formally tested sort of pruning-related mechanisms underlying depression. I think for me, I've always sort of, if I was going to go after that question, I think the question is like, where do you look, when do you look, what are your readouts, and we just hadn't gotten to that point yet. But I think now that, and also I don't know, we can go look in the genetics. I don't know if there's nothing, right? Yeah, there's no genetics, right?
Well,
there is some genetics.
Study with 200,000 people, but yet to be replicated.
Right. Yeah, but I think now that like this is suggesting this could be a more common pathway, it's with the right person who studies depression and knows where to look, and we have tools. Like we could start to ask, at least if the pathway is aberrantly regulated in the right place in the right time, and then you could start to knock it out and see if there's some rescue of different functions. But to my knowledge, it hasn't been done. At least, I don't think it's been done directly to test that question.
Wonderful. Thank you
Yeah.
so
Thank
much.
you.